Um, Thanks for joining us for this second video for this chapter. Um, the theme, of course, is DNA-related technologies. One of the main thing in this half of the video, I think it's going to be just one half, but we want to talk about is DNA cloning and related things. So clone, of course, is a copy, right? And what is DNA cloning entail? Now you're familiar with the chemistry of how you might amplify, make a bunch of copies of a particular segment of DNA, right? Once you do DNA cloning, you use that material to do either like sequencing and study it, or you could use it to do stuff. And in this latter half, we want to kind of talk about that doing stuff with DNA copies. That's what we want to focus on, particularly recombinant DNA technology, aka genetic engineering technology. So we're gonna use clone DNA to do things, interesting things. Well, the main thing we're going to do with it is to make proteins out of it based on the DNA segment. That's really the main thing. Now, that may not sound like a really exciting thing, but everything in biology revolves around protein, right? So, I mean, the sort of limitation, there's no limitation almost. There's no limit to like what we could do with proteins. So it's an exciting technology, but at the core of it is this technology that became available decades ago, which is restriction endonucleases. I mean, it's a mouthful, but often we would call it restriction enzyme or endonuclease enzyme. What does it do? It recognizes a particular sequence, specific sequence of DNA, usually a fairly short sequence, like 5 or 6 nucleotide long or something like that, or even 4 nucleotides, recognize a specific sequence and then cuts the DNA at that particular place. We won't refer to this very much, but there's also a group of enzymes called methylases that methylates, put the methyl group, CH3 group on DNA to protect that from being digested or cut from restriction endonucleases. If you wondered how in the world bacteria produces these endonucleases or restriction enzymes that cut DNA, well, they protect their own DNA with the methylases, right? Because even if the sequence is specific, like 5 nucleotide long or something, chances are if you have a lot of DNA, that 5 nucleotide sequence is going to just happen to occur somewhere, so you want to protect yourself. Because we found these enzymes, you could use both of them to make an interesting system where you could protect a certain part of certain DNA, but allow certain DNA in the same mixture to be cut, and there's a lot that you could do with that, but we're not going to talk about that much because that's pretty deep. But the restriction endonucleases, I want you to know how that works and how it plays into DNA cloning and heterologous expression of proteins. So these things cut DNA at a particular sequence, and you just have to remember that there's a complementary side to the DNA, right? Complementary strand. So let's say this, by the way, most endonucleases have a weird nickname like a 3 or 4 letter name plus number, kind of like Hind 3 or PST 1 or EcoR5, stuff like this. That's a typical name for a restriction endonuclease name. Usually ends with Roman numeral. Hind 3 recognizes this AAGCTT sequence, and it cuts right there. But on the complementary side, you have the TCGA. Now, these 4 nucleotide long space would be complementary to each other for the 2 sides that are cut, right? So what does that mean? I want you to remember this, that this is often called the sticky ends because and then it's not sticky as in like a universally sticky glue or something. It's only sticky for the complementary protruding end. right? So this endonuclease would only seek out the other protruding end left from the endonuclease's cutting or hydrolysis action. This is going to be really important later. So here's a PSD1 example. It cuts like that. There's also ones that leave a blunt protruding end, and there's ones that cut like into the 5' end, so that like you get a 3' protruding end, and different combinations of these, or different variations. These are the variations. Okay. Now, I guess we went over that already. Why is that useful? Well, because with this technology, we would be able to clone a piece of DNA into another DNA molecule. And we'll talk about why that would be useful in a sec. But here's how we might do this. Here's a circular big DNA. Here's a gene of interest. We could do, starting with the primer, right? We could do PCR amplification. Once you do, you got many copies of this, like exact same gene sequence, right? Now, if you digest it, that's kind of a jargon to mean you're going to apply a restriction endonuclease to cut it with, that leaves a sticky end, right? Here's the cartoon picture of a sticky end, right? And then if you cut this vector DNA, circular big DNA called plasmid, with the same endonuclease or restriction enzyme, like right here, you're going to get that same sticky end that's complementary to these sticky ends. So what can you do? Through a reaction called ligation, that's remaking the phosphate diester bond between like here and here, the two sticky ends together, so that it becomes a single DNA. In other words, we could insert this piece into that because they share the same sticky ends. Hope that makes sense. Now, why would you want to put your small gene of interest into a big circular plasmid DNA? Well, the reason is there's a fairly easy way to insert this plasmid DNA into bacterial cells like E. coli cells. Now, why would you do that? These bacteria cells are really easy to grow. If you could trick these cells into incorporating these recombinant plasmid, it's called recombinant because you combined sort of the natural-ish DNA with the sort of unnatural DNA that came from something different combined, and you made this like one single DNA from with sequences from a couple different places together. If you could insert this into an E. coli cell, and then if you could trick that E. coli cell into expressing or making mRNA based off this, and then the mRNA goes to the ribosome and it makes the protein based on it, you could trick this E. coli into mass producing the protein that is coded by this gene. Right? That's very helpful. And if you ever wondered how in the world we have like a commercially available amount of like proteins or enzymes, which are also proteins, this is how we usually put that inside E. coli and then trick it into mass producing that protein or enzyme of interest. Okay. Oh yeah, the sticky ends. So how does that go? Here's the process. And I want you to understand the process so that at the end of it, like in your own words and diagram, you could describe the process of DNA cloning in heterologous expression. What is heterologous expression? Well, I forgot to mention it in the previous slide, but the previous slide showed heterologous expression. Expression just means making proteins off of a gene. So that was making proteins off of that gene of interest, right? In a heterologous host, which means like host cell that's not you, that's not the gene of interests origin organism, if that makes sense. We'll come back to this. So how does this whole DNA cloning and generating a recombinant DNA and making recombinant protein, aka heterologous expression and all that, how does that all work? Well, it starts with a cloning vector. Here in the slide it says small. It's a fairly large molecule, but it's compared to like a genomic DNA that bacteria might carry. So the genomic, DNA would be the DNA that actually codes for how to make that bacterial cell. Plasmid, I forgot to mention, is a smaller piece of DNA that bacteria can pass back and forth between different cells, or sometimes even different species. Okay. Usually those plasmid, smaller DNA molecules, that's not the genomic DNA, is smaller and circular. Okay. That's not quite the same with eukaryotic chromosomal DNA. It's like a massively long chain. That's like how our DNA is stored and made or replicated. So let's imagine this is my DNA. This is bacterial plasmid vector. Let's say there's a protein in humans that we want to study. So you would mass produce it so you can study it. How would you go about that? Let's say this red part is that sequence in this chromosome that codes for that protein of interest. Maybe it's important in cancer biology. How do you do that? Remember, you already know how to design the primer that's going to be, or primers that are going to be used for PCR amplification. So you could amplify this gene, right? So, and then what you do after amplification is you cut the ends with restriction enzymes so that you get these sticky ends. With the two restriction enzymes, usually it's better to use two different restriction enzymes so that this end is different than that end. And then same thing with cutting the vector. Excuse me. You cut this often with two different restriction enzymes so that you know which end goes where. And then right, with these sticky-ended amplified gene, you mix that with the vector, and then you add an enzyme called DNA ligase that would remake the phosphate ester bond that was cleaved by restriction enzymes. I think I need a drink. I'm going to stop this video, come right back. Sorry about that. I'm back, and so on the last slide, I think we were talking about making recombinant DNA or plasmid DNA that's ligated with the DNA ligase at these two spots because of the sticky ends that were made by the same restriction enzymes at each end. And we earlier talked about this process transformation is a way to get these smaller circular DNA into E. coli or any sort of cells. That's called transformation in molecular biology or biochemistry. The way typically that's done, the easiest way to do it is you sort of heat the cell at a uncomfortable temperature, and at that temperature, right, like the reason why heat sort of kills us is because things start denaturing, and things start denaturing at high temperature because molecules start moving with higher kinetic energy, and so same thing happens to the cell membrane at a higher temperature. So if it gets more disorganized, and smaller DNA molecules like this has a chance to sort of slip into it, and because these bacteria somewhat rely on the information even in plasmids that they can pass along horizontally, right? These cells sort of allow that, and that's why this can happen. And then once you got this one cell with transformed plasmid, then you could grow this, and when it reproduces itself, it'll also make a copy of the plasmid that's inside. So um, a very popular organism for cloning, of course, is E. coli, just because its physiology is very well understood, and there's a lot of plasmids that have been developed or sort of discovered or well studied that we can commercially buy, and so that's why this is a really good deal. The plasmid itself, the construct of it, kind of often looks something like this, and at this point, like, I don't need you to know every single thing that's on this diagram, but it's actually not that much of an information. Remember, like, the restriction enzyme sort of cutting site sounds like these like weird acronyms, like ND1 or SAC2 right there, or SBF1. So any of these black bolded names, those are restriction endonuclease hydrolysis sites. Okay, and then in this circular DNA, and there's some other parts that we could identify that we'll talk about: the origin of replication, antibiotic resistance, and that's what this is. AMP-R stands for ampicillin resistance gene. Ampicillin is a beta-lactam compound like this. Remember, lactam is cyclic amide, and anything that's got that beta-lactam is usually targeted at the biosynthesis of cell wall in bacteria, and it shuts it down, and so the bacteria can't grow anymore, or it'll actually keep trying to grow, and as it tries to grow, when the cell wall synthesis is not keeping up, it just bursts in like the cell basically explodes. So that's that's how any sort of cellin type of antibiotics work: ampicillin, penicillin, stuff like that. But so why is that important? Well, you add this ampicillin resistance gene inside the plasmid besides the gene of interest that you're going to put in somewhere to make the recombinant plasmid, because that transformation process where you insert this plasmid into E. coli cells, that doesn't necessarily happen with every single cell that's present in the sample. And so if you just take any cell or cells out of that sample after the transformation and start growing it, you just don't know if the plasmid is actually in there or not, right? So what you do is you select for cells that have a certain characteristics, like possessing the ability to neutralize ampicillin. Actually, ampicillin resistance gene is a hydrolase that hydrolyzes this, so that the beta-lactam functional group is no longer there anymore. But that's with that, like you could select for cells that have that got the plasmid that you made, and this is how you would do it. You sort of streak the transformed cells, some transformed, some not, on an agar plate like this. It's made of agar, like this carbohydrate stuff that gives the structure and some food for the cells. And then on top of it, you add ampicillin in this solid agar material. So what happens is most bacterial, or really, most cells would just die, except for the cells that actually have this ampicillin resistance gene, and those can start replicating. And as they replicate, assuming there's not that many cells streaked on this plate, each cell is going to start its own colony, right? So that each of these dots is assumed to be started from like a single cell that started that colony. Sometimes you could add in sort of metabolic trick and some sugar molecules that are designed for this, so that the colonies that possess a certain plasmid is colored like this. That's another way to do it. But Anyway, we've already talked about the DNA ligase. Again, it sort of stitches together the 5' phosphate. Sorry, that's 3', 5' phosphate and 3' hydroxyl makes a phosphate ester to combine the two pieces of DNA. So it does that here, does that there as well. So now it's a completely intact double-strand DNA after DNA ligase does that. Now this doesn't really work unless you have the sticky end that are complementary to each other. Okay, and yeah, so the way you might design all of this is you need to make sure that there's a restriction site or restriction cleavage site here as well as on the other side. Like we said, usually it's better if the restriction enzyme is different on one end than the other, so you know which end goes in which direction, because direction always is important. In this case, you have this particular sticky end and a blunt end on the other. So as far as picking the plasmid vector, right, that you're going to use, it just kind of depends on whether it possesses all the necessary restriction enzyme site, right? And then the piece gene of interest that you're going to insert often comes from PCR reaction that's amplified, so you've got plenty of it to go into the vector. But before you insert, of course, you need to create a sticky ends on the two ends, and so you often have to sort of, the jargon is digest or subject this to restriction endonuclease reaction twice with two different restriction enzymes on the two ends. Okay, so here in this diagram it shows the same exact sequence on both ends, but that's usually not a good idea in practice, because then you don't know which side is gonna, you could insert the piece like this or like this, you don't know the direction, right? So typically we use two different restriction enzymes with two different restriction sequences on the two ends designed in, usually from like the primer and whatnot, right? So this diagram is not the best representation of what we do in practice because it's got the same sequence on two ends, but still this is like a good this is the multiple cloning site that they put into this vector. That this is actually a good thing to think about. So right, like they sort of design this into the plasmid vector, usually the ones you can buy, and the reason is in this multiple cloning site or MCS you have, like as the name suggests, multiple sites where different restriction enzymes can cut or cleave, and that's really helpful because this is where you sort of linearize that circular DNA and then insert the gene of interest. Like you could maybe cut it here with the HIND3 SMA1, and that's the two, that's this is where you insert the gene in this now circular DNA, like here and here, and then the your gene would be inserted here through ligation or something like that. Now, we've been talking about plasmid quite a bit, as opposed to sort of the bacterial chromosome or genomic DNA is much bigger. They're actually both circular in bacteria, but the actual bacterial genomic or chromosomal DNA in the nucleoid is much longer and bigger. Plasmid is much smaller. Usually in the cartoon it's shown like a perfect circle. It's actually not. Here's the electron microscope image of it. It is a circle, but it's all kinked and tangled and stuff like that typically. And here's a somewhat simplified diagram of a vector we might use. We mentioned origin of replication. That's usually programmed in there, and this is where has the sequence that signals to the bacteria that when it replicates, when it reproduces itself, it should also make a replica of this plasmid, not just the genomic DNA, and that replication begins here. So the polymerase, um, right? Well, the DNA has to be unraveled starting here, and then the DNA polymerase makes the copy and whatnot, right? And this is probably a more realistic diagram of a vector we might have. Is the origin of replication. It needs to be in there, otherwise it won't be replicated. So then there's no point in transforming the plasmid. Uh, and there's often some sort of operon operator sequences right before the spot where you're going to insert your gene. And remember the multiple cloning site. This site has a bunch of different restriction enzymes, cleavable sequences. So there's a whole bunch of them usually, so you could pick from multiple choices, right? And then you cut at 2 different sites here so that you could insert your gene of interest after ligation. And then often that is followed by a transcription termination sequence. So when the gene here is transcribed into messenger RNA, the the enzyme knows to stop right here, to stop producing the transcript or the mRNA right here. And then, of course, we talked about selection marker, like antibiotic resistance gene needs to be part of the whole plasmid. Okay, so that's what you look for when you're going to clone a gene of interest. Now, we talked about the operon sequences. Or at least I mentioned that we didn't talk about it. The purpose of that is to, this is the area or sequence right before the gene that sort of regulates how often and when, especially when, the gene that you're going to insert right here should get expressed. Remember, expression means the gene will be transcribed to mRNA, and then mRNA will be translated into an actual peptide or protein molecule. As you can imagine, right, like if we are expressing every single gene that can code for a protein in all over DNA, or in this case, I guess us as in bacteria, they're going to run out of energy, resources, and they're going to die, right? You don't want to just unregulated, random, infinite production of any and all proteins at all times. That's not wise, right? So there has to be a way to regulate when a particular protein should be produced. And there's different operon and a mechanism of regulation for different proteins that are sort of native to that organism, E. coli in our case. But we are inserting this like gene that doesn't belong in this vector, right, artificially. So we need a controllable way to sort of regulate that. And the way we do that often is called lac operon. And the way that works is a particular sequence that goes in before the gene of interest. Okay, and it has a sequence that's coded by this I, means inducer, but that this area of the operon sequence can code for repressor protein. Okay, so first repressor mRNA gets made and then translated into this repressor protein. What a repressor protein is it's designed sort of to bind to the operator part of the sequence right before the gene of interest that comes down here. And what happens is, right, that this protein prevents RNA polymerase from binding and producing transcript, so that whatever the gene that follows after all this sequence does not get expressed. It does not even get transcribed, right? And the we use this particular operon called the lac operon, and the reason why it's called the lac operon is because this actually is specific for when you have lactose present. Lactose, if you recall, is actually a disaccharide made of galactose and glucose, and when the bacteria senses that there's like some galactose coming from lactose around, it's designed to express the gene following, so that that gene codes for an enzyme that allows the bacteria to switch the fuel source to lactose, so they can start making use of lactose and get energy out of it instead of some other sources of nutrient. And so it's sensitive to the presence of like galactose. And when you put in something like galactose or lactose in the culture, what happens is that's going to sort of inactivate the repressor protein, so that this comes off and then it gets inactivated. Then what happens is the gene here, the galactosidase, that can help with the lactose digestion, or I should say metabolism, can be transcribed and expressed and start using lactose as a fuel source. Now, so usually, right, this inducer, or what inactivates the repressor, is this lactose molecule that has the galactose and glucose in it. But this lactose can complicate things in a culture. So what we do is a sort of artificial molecule that looks like lactose, but not quite, but still can bind to the repressor, so that it stops the repressor action, so that the gene following down here can get expressed. And so so that's a nice discovery, that this artificial lactose mimic compound can do that. So what we do with the cloning is we copy this part of the regulatory operon sequence up here, and then put that in front of the multiple cloning sites, so that the repressor would actually be in operation, usually blocking the transcription of the gene following. But when you add IPTG, the repressor gets inactivated, so that the transcription and translation of the following sequence would resume or start. Okay, and so it's kind of a long-winded explanation, but lac operon responds to IPTG and allows the transcription or the expression of the gene that follows after the lac operon sequence. Hope that makes sense. And that's something usually what's in the vector. So this is what we mean by, I think earlier I talked about you can trick your E. coli cell into mass producing your protein of interest. And if you add enough IPTG, you completely inactivate the repressor, so the E. coli gets busy expressing this gene right here. And that's what we mean by tricking the cell. And now what I want you to do in class, maybe we can work on this together. Your partner, I guess, would be your teammate. See if you could summarize all this information essentially into a diagram and design an experiment to clone a gene from your DNA. So you need to think about how you could PC amplify it, and then put a piece of gene into E. coli, a plasmid vector, transform it into E. coli, and then have the E. coli express that protein that's coded by your gene. And what do you need, right? Like how, and then what are the procedures? If you understood everything we talked about in this latter half of the chapter 9 video, you should be able to do it. So